Hi everyone. So, as many of you probably know, Sauropterygia was a highly abundant and long-lived clade of Mesozoic marine reptiles. In this project, we focused on understanding how axial body plan changed in the clade through their 180-year million history. So first of all, I'm going to give you a brief overview of the clade, then I'm going to explain some important terminology for the rest of the presentation, and then go into details of the methods that I used and the results that we got. So the first Sauropterygian fossils appear in the earliest Triassic. Initial radiations were in shallow water nearshore environments. You get things like the armored placoodonts, pachypleurosaurs, nothosaurs, and pistosaurs. After a long period of decline throughout the late Triassic, possibly as few as one lineage crossed into the Jurassic. Once this happens, we get an open water radiation. This is where we see the appearance of these classic plesiosaurian morphotypes, so the long-necked plesiosauromorphs and the short-necked pliosauromorphs, which convergently evolve repeatedly throughout the rest of their evolutionary history. So some Jurassic taxa, some of which survive into the Cretaceous, are things like mammaliasaurs, pliosaurs, cryptocleidids, and microcleidids. After a protracted period of high turnover across the Jurassic-Cretaceous boundary, we get the radiation of the leptocleidids and extremely long-necked elasmosaurids in the Cretaceous. So this huge morphological variety in axial body plan represents an excellent opportunity to quantitatively assess macroevolutionary change with an aim to perhaps understanding the mechanisms of this change. The reason Sauropterygians are particularly suitable for this is we've got a huge range of cervical vertebral counts, between 6 and 76 in this data set. As I said, they persist for the whole of the Mesozoic, and also the adult morphology corresponds in a relatively direct way to certain developmental processes. So onto some terminology. What are those important developmental processes that can affect axial body plan evolution? So this is the pre-Semitic mesoderm. It's what eventually forms the spine and associated structures in vertebrates. So the first important developmental process is Hox Gene Signaling. So this basically controls, depending on the region that a somite is in, whether it becomes a cervical or a dorsal vertebrae. Once the identity has been set by that Hox Gene Signaling, we get the effects of the Semitic clock. Now this basically controls how many somites we have. If the clock ticks quickly, we get a relatively high number of small somites. If it ticks slowly, we get a smaller number of larger somites. Once the identity and number have been set, it's then possible to get differential growth of somites. So this might be that some of them grow larger than others. This is what you get in long-necked mammals like giraffes and camels. So how do those developmental processes actually correspond to the evolution of adult morphologies? Well, if we consider a hypothetical pre-sacral series with cervicals in yellow and dorsals in purple, so we've got 3 cervicals and 5 dorsals to begin with, changes in Hox genes, so homeotic effects, here we've got one dorsal has switched to a cervical. Semitogenesis, so this is changes in the rate of the semitogenetic clock. We've got a higher number of cervicals and a higher number of dorsals. The clock has ticked faster. And then finally, differential Semitic growth. Hopefully you can see here that the yellow cervicals are now longer than the purple dorsals, on average. So, with all of this information in hand, we wanted to test 3 explicit hypotheses about the evolution of axial body plan in Sauropterygia. So the first of these was that the combination of homeotic effects and semitogenesis was more important for axial body plan change than differential growth. The second of these was that homeotic effects and changes in semitogenesis were decoupled across the phylogeny of Sauropterygia, and this was based on some previous work that had been done that showed that these 2 things were decoupled across all amniotes. And finally, we were interested in whether and finally we hypothesized that homeotic effects were the more important driver of axial body plan change as opposed to semitogenesis, so the addition or subtraction of vertebrae. So to test these hypotheses, we collected a large data set of cervical and dorsal counts and series lengths from museum specimens. This included over 80 taxa, covering all major Sauropterygian families. We also used a phylogeny. This was a composite tree, so Jurassic and Cretaceous taxa were from a new cladistically inferred topology, and Triassic taxa were from a cladistically inferred topology from Jiang et al. 2014. We got the ranges, the occurrence data from the paleobiology database, as well as some other publications. We used all this information to generate 100 timescale trees to take into consideration the effect of variation in the topology and the possible error and range data. So when I get to the results, the statistics that I will report are the average results over those 100 trees. So we use that data to collect a set of 4 measurements. So again, we've got our hypothetical presacral series here. We measured the overall length ratio, so this is the ratio of the length of the cervical series relative to the dorsal series, so the total length of yellows to total length of purples. The unit length ratio, which is the length of an average yellow cervical relative to an average purple dorsal. The count ratio, which is just the number of yellow cervicals relative to the number of purple dorsals, and the presacral count, which is the total number of presacral vertebrae, so cervicals plus dorsals. We took these measurements and we analysed them using 2 different phylogenetic comparative methods. The first one was Auteur, which is a Bayesian method that can be implemented using the Geiger package in R. I think this has been discussed already, but basically this optimizes rates of continuous trait evolution across the branches of a phylogeny whilst reducing model complexity. The second thing we did was take standardized phylogenetic independent contrasts. This is quite a common method that many of you are probably familiar with, that's often used to correct for shared evolutionary history between taxa. But because it shows the magnitude and direction of trait change across the nodes of phylogenies, it can be used to, to indicate the positions of rate shifts on a tree. So, to move on to the results, this is the output of the Auteur analysis. So this is when we were trying to address hypothesis 1. So this is whether the combination of homeotic effects and changing semitogenesis was more important than differential growth in terms of the overall axial body plan of Sauropterygian taxa. So to do this, we compared the overall length ratio rates to the count ratio rates. And you can see from this, there is a certain amount of correspondence between these trees. So we've got lower rates in Triassic taxa at the bottom, Placodonts and the Pachypleurosaurs, a shift to slightly higher rates in the top of the tree. But there are also some places where there are differences. So, for example, we've got slowing down in the pliosaurs and in the length ratio, and a much higher rate increase in elasmosaurs and leptopilots. So it seems that count ratio probably is influencing length ratio evolution to a certain extent, but there is some variation around that. So to unpick this a bit further, as I said, we use standardized independent contrasts. So when we do a regression of length ratio contrasts against count ratio contrasts, there is a strong correlation with an R-squared of 0.73. So this indicates that homeotic and semitogenetic effects are driving evolutionary change in the length ratio, so evolutionary change in axial body plan. But we also found that when we did a regression of length ratio against unit length ratio, so this is the influence of differential semitic growth, we also got a significant result. This R-squared was much lower, of 0.25, but that is still having an influence. Moving on to hypotheses 2 and 3, which are probably best considered together. So this was whether homeotic effects and semitogenetic change are decoupled across the tree, and whether homeotic effects are more important than semitogenesis for driving axial body plan evolution. So again, we have the Auteur outputs here. We compared count ratio rates with presacral count rates. And here, again, there's a small amount of correspondence between the trees. So There's higher rates in the pistosaurs and the microclidids, but we've got some important differences here as well. So the presacral count rates are much higher at the bottom of the tree, for example, and the leptoclidids at the top slow down a lot more. So it looks like, in the first instance, that these two things could be decoupled. So again, we did phylogenetic independent contrast analysis. And what we found when we did a regression of count ratio contrasts against presacral count contrasts was that they are actually significantly correlated across the tree, but with a very low R-squared of 0.17. This is contrary to the previous result across all amniotes, which suggested that these two things are completely decoupled across the tree, but it's still a very low R-squared. Then we went into a bit more detail, and we looked at the standardized contrasts that corresponded to nodes that were on the backbone of the topology, and separated from those contrasts that corresponded to nodes that were within nested clades within Sauropterygia. When we did this, what we actually found was that the backbone nodes had a much better correlation, significant correlation, with presacral count contrasts, and the nested nodes had no correlation at all. So this suggests two things. The first is that, in fact, homeotic effects and somatogenetic change can be correlated across the phylogeny, but they weren't in the nested subclades. And in terms of what the important drivers are of axial body plan evolution here, it suggests that normal background evolutionary change in axial body plan is driven by homeotic effects. But at the origins of major subclades within Sauropterygia, so along the backbone of the tree, somatogenesis was coupled with homeotic effects and was also an important influence on axial body plan change. So just to summarize everything, the first hypothesis was that homeotic effects and somatogenesis were more important than differential growth in Sauropterygian axial body plan evolution. We found that they were more important, but differential growth also did play a role. The second, that homeotic effects and somatogenesis were decoupled across the tree. We found that in fact they're not, but if you separate them out into nested and unnested parts of the tree, the nested subclades within Sauropterygia do show decoupling. Finally, we found that homeotic effects are the major driver of normal kind of background patterns of axial body plan evolution within the clade, but during periods of major reorganization of the axial body plan at the origins of major subclades, major subgroups within Sauropterygia, somatogenesis is an important contributor to changes in axial body plan. So finally, the kind of main message to take away from this is that heterogeneous statistical models can be extremely important in terms of unpicking the actual mechanisms that are driving evolutionary body plan change in vertebrates. Thank you.